Hello, I'm Robert LeBoyer, Senior Biotechnology Analyst at Noble Capital. With me here today is Dr. Shankar Mansunuri, the CEO of Ocugen. Ocugen has been developing gene therapies for diseases of the eye and has 3 products in clinical trials. To begin with, can you tell us a little bit about gene therapy in general? I know that there have been several products in development. There have been some successes and some products that have been that have reached the market, but they're limited markets, very expensive products. So could you just differentiate your approach and technology from things we may have heard in the past?
Yeah, absolutely. Robert, first of all, thank you for having me today. Gene therapies as a whole, we all agree they are greatest discoveries of 21st century. They can do a lot of good for humankind. So, however, traditional gene therapies, if you have a defective gene, you try to give a functioning gene. The defective gene is still there, and it's producing whatever misfolded protein, and you still have that. So what is important is genes don't work in isolation. They work as a network. So one has to look into, based on epigenetics, a lot of data is coming out. One has to look into the system and the genetic network. For example, in retinal space, what we found out, when you have any defect and you have key functioning genes, which are responsible for certain functions, such as cell development, metabolism, inflammation, and phototransduction to cell survival. Some of these key functioning genes are depressed. And then there's a missing link to the master regulator. That's a modifier gene. So that's what we work on. We don't work on a specific gene. We work on the entire disease. So what it means is once you upregulate that modifier gene, which is master regulator, and all these key functioning genes which are depressed, they upregulate and they reset the homeostasis and create a healthy environment for cells to survive. That's a very powerful concept because retinal cells, they're non-dividing cells. What we have or what we have for life. And if I can do something to reset them and create a healthy environment, potentially they should survive the rest of your life. So that's very important distinction between us and other gene therapy approaches. The approach is good, but you still have a defective gene with traditional gene therapy and you're giving a functioning gene. In some cases, for example, photoreceptors, they're degenerating and they're not doing anything to stop that. And that's why in some cases, you're not getting results you anticipate. Some of the clinical trials are failing, even in the retinal space. So you're assuming the system is okay. The system is not okay. What we go after is entire disease. We're trying to fix the network and create a healthy environment for cells to survive. That's very important. So that's why we differentiate ourselves from traditional gene therapies, which go after a very specific genetic defect. In some cases, you have 50 patients, few hundred patients, maybe even 2,000, 3,000 patients. Commercially, in the long term, how is it going to be viable? A lot of gene therapy companies, early companies who started launching, they had a good technical success. They're doing a lot of good for these patients, but the population is not huge. So from commercial terms, the cost is also very high. Some of them started charging $3 to $4 million. Even then, if you only have even 1,000 patients, how long are you going to run? So from shareholder perspective and from commercial viability perspective, and that becomes very difficult for people to understand. Wow, it's a great product, but they're not making a lot of money. Unfortunately, people look at from that scope. We spend so much time, 8 to 10 years, to bring each product to the market. And at the end of it, you need to have commercial success for the technology to move forward. So those are the differentiating factors. What we work on, first of all, entire disease. We're not working on 50 patients. We're not targeting 1,000 patients. We're targeting hundreds of thousands of patients to millions with our gene therapies because these are modifier platform. So they work on the entire disease, not a specific defect.
I see. And you just mentioned the idea of a gene network and the idea that some of the gene therapies will add a gene to a person with a disease, but not eliminate the defective gene that's there. And your approach is to modify the gene with the master regulatory gene and affect the entire downstream network.
That's right. Yes.
Okay. And one of the things you mentioned was the populations. So could you talk a little bit about the clinical trials that you have going and the different products, how they work, the markets they serve, and where you are in development for them?
Yes, absolutely. I'll start with our first program, which is in the late stage, targeting retinitis pigmentosa. Retinitis pigmentosa is a conglomerate of inherited retinal diseases, or defects in over 100 genes can cause RP. There's about 1.6 million people globally struggling with it. And between US and EU together, we have about 200,000 patients. And that's a large population. And when you look into that, 200,000 to 300,000 US and EU, currently there is only one therapy available. I said there are over 100 genes, defects in 100 genes can cause the disease. There is only one product available, which was first gene therapy in 2017. And that affects RPE65 gene. And that has probably 1% of the entire RP population. So in US itself, we have 110,000 patients. So majority of the population don't have anything. They're really desperate. And the disease is very progressive. You will have peripheral vision loss, night vision loss. Eventually, you're left with central vision. Eventually, that goes away. And there is a patient video on our website, elderly patients, and he lost his peripheral vision, night vision. And now, with our gene therapy, he's not only able to control disease progression, he's actually getting his peripheral vision back. And so that's a phenomenal effect. And some of the patients, once again, depending on where the disease is, our gene therapy in phase 3 clinical trial is targeting not only entire RP as a disease, irrespective of what mutations you have, syndromic, non-syndromic. It's also targeting patients from early stage of the disease to late stage of the disease, from pediatric population 5+. So it covers entire RP population, most of the RP population. That's very important. So you're targeting between US and EU, let's say about 300,000 patients. You're not targeting 1,000 patients or 2,000. So we will have a lot of flexibility with pricing. Number two, safety. Some of the gene therapies out in the market and systemic gene therapies, a lot of times you see, significant adverse events. And these are severe SAEs, you can call them severe adverse events. And sometimes, they lead to death too. And so systemic gene therapies and retinal gene therapies, there's a big distinction. Systemic gene therapies are not targeted. That's why they have to give 1,000 to 10,000-fold higher dose compared to our gene therapies. We go directly into retina. It's a subretinal surgery, and it's not a complex one. They do it routinely. And so that's done, one and done. And it's very specific and targeted. So therefore, we can get away with a much smaller dose, which could be very effective. So that's it. Because of that, we are seeing so far in all our clinical trials, 3 programs, as you talked about, we don't have any SAEs related to the drug, which is very important, safety. And so, again, recapping on RP, about 300,000 patients in US and EU, globally, many more. And currently, there are no approved therapies except for one small target population within that group. And we are targeting that large group with our phase 3 clinical trial. We are currently, it's a large study. It's the largest orphan gene therapy trial in the world, 150 patients. And we're planning to complete recruitment in the next few months. And that means the final data, it's a 1-year duration. We have to follow these patients. And that will be done by mid next year. And they're ready for BLA and market authorization in Europe. And the good thing is, based on the data they have seen from phase 1, 2, and also unmet medical need, EMA has agreed. that one clinical trial we're doing in US is good for them for approval. So we don't have to do any additional clinical trials in Europe for approval. So we are targeting parallel BLA and MAA submissions. We also have a regenerative medicine advanced therapy designation in the US for our RP OCU400 program that will allow us to initiate rolling submission. So a little later part of this year, after we are done with our recruitment, we're going to engage with FDA and discuss the path forward on the rolling submission on the timeline. Potentially, we can start doing that late this year, rolling submission. That means, we'll start with the different sections, non-clinical, CMC, and manufacturing. We are on target to complete our process validation runs this year, which are required for BLA submission. So that's on target. And so that means that section can go before final clinical section comes in. So once you lock in the clinical section, after the last patient comes in, sometime next year, the BLA package is complete, and then you have 6 months to get approval based on the accelerated path, regulatory path. So that's the plan. So RP, the first foremost, what are the inflection points for the program OCU400 this year? Completion of the recruitment, which is a big milestone. We are keeping tabs on it, and the recruitment is going well. And we're going to let the markets know as soon as we're done with the recruitment this year. The second step is working with FDA and initiating the rolling submission. And we're going to let the markets know. So that's going to tell you we're on target for next year completing BLA and MA packages, and also get ready for approval and launch early 2027. So everything is on target. The second program, Stargardt, another inherited retinal disease, and it's a central vision. You get atrophic region, and you start doing, instead of going from peripheral, it starts with the central vision. And the disease progression is very similar to dry AMD geographic atrophy. There are multiple pathways which can cause disease progression, starting with oxidative stress, lipid metabolism, which are the core factors, then inflammation and complement system. And so we have another modifier gene, Aurora, which regulates all these 4 pathways and also create a healthy environment for retinal cells, photoreceptors, and RPA layer cells to survive. That's very important. So currently, Stargardt disease has about 100,000 patients within US and EU. So currently, there are no approved therapies. Also, the ABCA4 defect causes Stargardt disease. It's a very large gene. It's very difficult to package that in a single capsid using traditional gene therapy to deliver. That's why no other companies have made attempts to progress. There may be other therapies available targeting some of those pathways to slow down the disease progression, especially the oxidative pathway, which is critical. However, nobody has made a progress where we are going with it, with a one-time treatment for life. Recently, as we announced, we had a very good success with FDA based on our phase 1/2 data. We worked with them, and we requested, because of the unmet medical need and very promising data we are seeing in phase 1, we can convert our phase 2 into phase 2/3 pivotal confirmatory trial for approval. And FDA has agreed. That means we knocked off phase 3. The traditional approach is go to phase 2, then go to phase 3. That will cost you additional 2 to 3 years and significant resources, including capital. By knocking that off, patients will have access to this medicine potentially 2 to 3 years ahead of time. So that program, where do we stand? We had a good agreement with FDA, and we are about to embark onto our phase 2/3 pivotal confirmatory trial within the next few weeks. We're working with FDA, and as soon as our protocol is final, our IND amendment is cleared, we're going to start initiating the trial. So once again, this has got a 1-year duration. We have to follow the patients, and our primary endpoint in that going to be measuring that atrophic lesion size, and the secondary is going to be visual acuity, which is a functional endpoint. And that means if you give 9 to 12 months for recruitment, about 1-year duration to follow up, we'll be ready to file BLA in 2027. Similar to RP, because of unmet medical need, currently we are working with EMA, requesting them, can we use this 51 patient trial in US for submission of market authorization EU? So we will know the result of that in the next few months. And so that means if they agree to that, this will be very good for the patients. We'll be able to complete both market authorization application in Europe and BLA in 2027. So the last program is geographic atrophy, which is a late-stage dry age-related macular degeneration. That's OCU410. And there's a significant unmet medical need because geographic atrophy is very debilitating disease. I mean, you could be in 65, 70, and you've done everything in your life. This is the time you really have to enjoy. And now suddenly your central vision, you have, atrophy. That means central vision gets blurred and you cannot read, you cannot write, you cannot drive. And some people, they want to spend time with their grandkids and it's making them debilitated. They cannot do the things they want to do. They're dependent. And it's really very difficult if you put yourself in patient shoes. And FDA, about a year ago, approved 2 products in the market, and a Syfovre and another product. These products target one of those pathways I talked about. Remember, the disease pathogenesis, how it progresses significantly, is because of oxidative stress and lipid metabolism. As we age, there are certain nutrients, you need it in a certain form. There are functioning lipids like lipofuscin in retina, and they need those nutrients. Once you don't get them in right form, they can start aggregating. And you got a RPE layer, which clears all the debris. And when it gets clogged up, you get an inflammation. And then eventually you form the complement system. So the pathogenesis, if you understand the disease progression, what are the key factors? You really have to control oxidative stress and lipid metabolism. And this gene, as I mentioned, regulates all those 4 pathways, RORA, and it's the same gene which we are using for Stargardt disease, OCU410ST program with a different dose. And this is showing a promising results so far in phase 1. And we have already started phase 2. In fact, first quarter of this year, we announced the 50+ patient trial is on track. And in fact, we recruited all these patients in a record time, ahead of time. Because the current therapies, you require every month or every other month injections intravitreally in the eye. And they do have a low to moderate efficacy, reducing the lesion growth by about 30% in 2 years. However, they do have a significant adverse events. 10 to 12% of the patients may get wet macular degeneration for treatment with dry. Even then, these products were approved because of the significant unmet medical need. However, Europe, EMA didn't approve any of these products because they felt they didn't show any functional benefit to the patients. In our phase 1 trial, as early as 6 months, we're seeing significant results. Not only significant control of the atrophic lesion growth, and also tissue preservation compared to better than, products in the market. It is also showing significant benefit in visual acuity compared to untreated eyes. So we are seeing a significant functional benefit in these patients. So these results are very promising, even though preliminary. And currently, the phase 2 trial, which is successfully ongoing, and we're anticipating in the next few months interim results from that. And
Right.
looking, structural, which is atrophic region, as well as functional visual acuity results. So based on that interim results, we're going to work with FDA and DMA and finalize our phase 3 design for the clinical trial. By the time we're done with phase 2, by first quarter of next year, our goal is to get ready for phase 3 with the agreement with FDA and DMA on the phase 3 clinical trial. So this is going to be remarkable. I mean, imagine, there are therapies out there, reducing the progression of the disease by a bit at the long-term use, and also every month or every other month. And you have something which can really remarkably change the way you treat the disease. Just as other gene therapy programs, our gene therapies actually create a healthy environment for retinal cells to survive. It's a powerful concept. So not only in some patients, you may be able to not only control the disease progression, you may be able to stop it, or you may be able to even reverse it. These genes have potential to do that. And with a single potential injection for life, that's it. It's a one-time therapy. That's going to change the paradigm shift. It could become a gold standard. Why? This is a huge disease burden. More than 200 million people struggle with, dry age-related macular degeneration. And if you look at US itself, and if you take US and EU together, you got 2 to 3 million patients with a late stage geographic atrophy. So this is a huge disease burden. We can do a lot of good if you're successful with this program. I mean, all these programs are remarkable, combining retinitis pigmentosa, Stargardt, which are the major inherited retinal diseases with the vision loss. And then the big one is dry age-related macular degeneration for elderly, for late stage. And now, if you combine all these programs, they should encompass majority of the blindness diseases today, which you can quote them as significant unmet medical needs. And with these 3 programs, our goal is to file all 3 BLA MA filings in the next 3 years. 26, retinitis pigmentosa, OCU400. 27, OCU410ST, Stargardt disease. 2028, geographic atrophy, OCU410. In 3 BLAs in the next 3 years, targeting majority of the blindness diseases. So that means Ocugen, we already believe, we're confident, we're leading ophthalmology gene therapies. And in the next 3 years, with those regulatory filings, we'll be able to bring these products in the next 3 to 5 years to the market. And that'll be a big, big support for all these patients. And they don't have anything today. A lot of them have, they're desperate for hope, preserving their vision. As we are talking today, many of them probably are becoming blind, legally blind, because of lack of therapies. And that's the impact we can have in this patient population globally.
Great. That's really remarkable. 3 programs that will have BLAs in 3 consecutive years is outstanding and very rare to see. Just to reiterate some of the points that you raised, in terms of the operation itself and using these drugs, in the investor day where you had a panel of the doctors who'd been treated and some of the patients, one of the things that came out was the treatment. And you mentioned 1 treatment for life. And one of the things the doctors brought out was that these procedures to use these drugs are already standards of practice. So there's no training involved. This is something where any ophthalmologist could use the product right from the start. And it doesn't require any special training or procedures, just one and done.
Yes.
And one of the other things was that the patients actually said that their vision is being restored. So one of the questions was, well, how is that happening? It's very common to see a slowing of progression, but actual reversing symptoms is unusual. And the answer was that the master regulatory gene approach restores homeostasis and the normal function of these genes in the pathway so that it restores vision as well as slowing the progression. Is that an accurate summary?
Yes. Yeah. That's an accurate summary, because you're working on the entire network of genes and resetting the homeostasis and restoring the function, and also creating healthy environment for cells to survive. That means potentially with one-time treatment, they should survive the rest of your life. And you have seen the data, long-term data. We're looking at 2-year data came out from low light visual acuity in our OCU400 RP study. And we had evaluable subjects. 10 subjects were evaluable at 2 years. And the data is remarkable with a p-value of 0.005. I mean, that's compared to untreated eyes. So the effect was actually smaller, even though it's still under p-value of 0.05 at 1 year. It improved further. That means the benefit of treatment is improving as the time goes on. Again, validating the durability.
The durability and the the actual concepts underlying
Yes.
the treatment, that this is restoring homeostasis, it's one treatment necessary, and it's durable. So in terms of the actual endpoints, you mentioned one structural and one functional. So is that consistent with the FDA's thinking at this point? And the actual measurements are something that people might not be familiar with. Could you just touch on what's being measured to determine the functional improvements?
Yeah, absolutely. Based on the disease, they differ. For retinitis pigmentosa, because patients lose their peripheral vision and night vision first, then the central vision is left. So the first product got approved using a mobility test. And so that's a good way of functioning. If I can walk in a low light, in my house, going to the kitchen, able to do my things, or you go to a restaurant without any help, I can walk to my chair and sit. A lot of these things are practical things. I can do my things on my own with limited help. So how do I do that? So there is a mobility test, and it goes from bright light outside to low, dull moonlight. And in fact, FDA, I should really thank them. After the phase 1 to clinical trial, they worked with us and made it, increase. In fact, implemented more levels instead of stopping at the moonlight. We went to dull moonlight. Why? Because that will allow us to get more patients in the clinical trial. So if we can show we are improving 2 levels, that means they can walk through this maze with the different light conditions. Today, I'm walking at certain light level, brighter light level. Then I can walk after the treatment after 1 year at 2 light levels lower than that. And that means I'm improving, right? Because
Yes.
the reason this course has obstacles, so that means it's considering 2 things, like looking at, overall functioning, how I'm functioning when I'm walking through, and also with the different obstacles and different low light conditions. So all of us, as we age, right, we have low light vision issues, right? So therefore, compared to BCVA, best corrected visual acuity, low light visual acuity is more sensitive in a shorter term to measure. That's why we use that for these patients. So in this mobility test, that's a functional test. Then the secondary measure is low light visual acuity in these patients. So because RP as a disease, this is more reasonable and practical for them to measure. That's what first FDA approved the product. So that's what we adopted. In the sense, it's our own proprietary course. We worked with FDA, made it more specific and sensitive compared to what other company has used to get approval. The second program and third program, Stargardt disease and GA, we're looking at the central atrophic region. So that's structural. And so if you're trying to measure within a certain time frame, like a 1 year, that's a good measure to see the disease progression, how it's doing, treated versus untreated. And so that's why FDA believes that's a good measure, structural as a primary. However, functional benefit is very important. So in Stargardt, we agreed the functional will be the secondary measure, what we are already seeing a good progress. And that's what we are using. That's what we agreed with FDA. We believe FDA will probably do the same with geographic atrophy based on our discussions on Stargardt. So use the structural as a primary and use the visual acuity as a secondary measure, because structural will be able to catch the signal within 1 year time frame. We also have to be mindful of efficiency of clinical trial when there is so much of unmet medical need. Yeah, if you run clinical trials for 3 years, 4 years, sure, you can look at some other measures, but so that's very good. And thanks to FDA, they understand the limitations, they understand the unmet medical need. So I believe that they're going to be, based on our experience at Stargardt, GA, they may agree to that. Again, we also have to work with EMA, and time will tell. Hopefully, they will agree with FDA's assessment. And as long as they're seeing functional benefit as a secondary in Stargardt and GA, they may be fine, because you have to show some functional benefit. That's really important.
Yes, absolutely. And one of the things that one of the consequences of losing vision is trips and falls. Yes. And the need for custodial care. So by doing this, you're not only benefiting the patients, but you're also saving cost of caring for these patients.
That's a very good thought, Robert. I mean, a lot of times, yeah, I mean, if you're in patient shoes, you really understand what they go through. I mean, all these
Yeah.
diseases are debilitating. At some part of my life, I'm thinking if I'm a patient, oh my God, I'm going to be legally blind. And so how is my family going to treat? It's a family, there's a cost, and also they have to go through so much. And there is societal cost. There's a, cost from healthcare. And if you put all this together and the treatment, if you're able to rescue somebody from becoming legally blind, and the value for that is invaluable, you cannot measure it quantitatively.
Yes,
That's the hope. With our gene therapies in the next 3 years, we're planning to file, and we want to make a remarkable impact globally in these patient populations who are really desperate today with these blindness diseases.
yes, absolutely. And speaking of impact, could you just reiterate some of the milestones for the trials going forward for the next few months?
Absolutely. First and foremost, OCU400 targeting RP, we're planning to complete recruitment and also initiate rolling submission this year. Those are the two milestones. For OCU410 Stargardt disease, we got agreement with FDA. We're about to embark on our phase 2/3 pivotal confirmatory trial. As soon as we initiate the trial, and we're going to let the markets know, and we'll also continue to release. We already released partial data from our phase 1 trial. We'll release the complete data in the second half of this year when most of these patients come through 12 months. And also, last program, OCU410, targeting geographic atrophy. We are completing our phase 2 right now. We are definitely going to release in the next few months interim data from the phase 2 in many patients. In addition to that, we're also going to release our 12-month data from phase 1. So there are a lot of data points coming up in the second half. And so
Yes, these are many data points from late-stage trials that not only show experimental verification of the concept, but the real clinical data that you can use to file. So
yes,
this is great news, and I'll be looking forward to following these milestones and seeing how things work out.
Yes, thank you, Robert. That's
Okay.
hope. We have a lot of milestones coming up this year and next year, and the goal is to continue to my team, and thanks to my team, all our investigators and patients, and they work every day and night very, very hard, because it's not easy for a small biotech to launch 3 BLA and market authorizations in the next 3 years. It's a lot of work ahead of us, but we're very excited, and we're motivated with the data we are seeing in the clinic. We can do a lot of impact, and so that's what drives us.
When I first heard of the concept of master gene regulatory of that approach, I thought, wow, this could be a breakthrough. And now seeing these things in late stage clinical trials with BLAs on the horizon is really exciting. So congratulations on all the progress. And I look forward to seeing the announcements of this data.
Thank you, Robert. Thank you for having me again today.
Well, thank you for speaking with us and letting us know about these details on the clinical trial.